Welcome to the Beer Taster's Notebook, the show focused on beer education. I'm Jonathan Thompson, Advanced Cicerone, BJCP beer judge, and fermentation enthusiast. Welcome back to the Beer Taster's Notebook, episode 21. So before we jump into yeast, let's talk about fermentation and cellular respiration in general. I think this is all just really good to know because it helps to understand what the yeast is doing, and then we understand sort of secondary products and such things. So the bottom line here is all cells need energy to survive and to perform their various functions. I think that should be pretty clear. So adenosine triphosphate, ATP, it's the primary form of energy. And in order to build these units of ATP, cells perform a process called cellular respiration. And so we'll get to all this in a minute, but generally this process involves glycolysis, which glucose splits into pyruvate, which then enters the Krebs cycle, also called the citric acid cycle, also called the TCA. And this is where a bunch of stuff happens, and then electron carriers help with step 3, and this is called the electron transport chain, and this is where oxygen is employed, which is why we need to breathe, and why if we can't transport enough oxygen, let's say through our blood via issues with hemoglobin or whatnot, then, bad stuff happens. And so all of this stuff happens in an aerobic situation, so, in the presence of oxygen. And I just note that oxygen is the primary electron acceptor at the end of most of these chains, but there are substitutes in certain situations. But let's say there's no oxygen available, there's no substitute, and then, in that case, a different process called fermentation takes place. And, of course, this is our liquid bread and butter, so to speak. And so let's first deal with the glycolysis. It's pretty simple. It requires no oxygen and involves 10 steps, really, but we can split it into 2 groups. So the first 5 steps involve spending energy, and this happens in the cytoplasm, by the way. And then the second stage generates the energy that we're looking for. So let's take a look here. Okay, so step 1, an enzyme called hexokinase pulls a phosphate group from ATP, transfers this to a glucose molecule, and now we have glucose 6-phosphate. Step 2, the glucose 6-phosphate gets isomerized by phosphoglucose isomerase, another enzyme, into fructose 6-phosphate. Then step 3, another enzyme uses ATP to convert the fructose 6-phosphate into fructose 1,6-biphosphate. And for step 4, this fructose 1,6-biphosphate is cleaved into 2 molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and this uses the enzyme aldolase. And so step 5, the dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate by triose phosphate isomerase, and that's the end of the energy spending phase. Okay, now step 6, and this is where we get some energy back. And more, actually. So the glyceraldehyde 3-phosphate is isomerized and phosphorylated into 1,3-biphosphoglycerate and NADH. Okay, so then the enzyme responsible for this it's known as GAPDH because its birth name is glyceraldehyde 3-phosphate dehydrogenase. So it's just easier to go by the acronyms a lot of times. And so now step 7, we're getting some more ATP back. The enzyme phosphoglycerate kinase moves a phosphate from the 1,3-biphosphoglycerate into ADP, turning it to ATP, and then leaving 3-phosphoglycerate as a byproduct, or as I should say, is just as a product. And then, step 5 rearranges the 3-PG. We're just going to start using acronyms now a little bit to make 2-PG, and this is done by phosphoglycerate mutase. Step 9 sees water being removed from the 2-PG by enolase, and now we are left with phosphoenol pyruvate. Okay, now the final step, this phosphoenol pyruvate has a phosphate removed by pyruvate kinase and transferred to ADP, and now we have ATP and pyruvate. So recall that in phase 1, we ended up with 2 GAP molecules, and so step 2 generates 2 ATP per, but there's a 4 in total. So we get a net gain from glycolysis is 2, and there we go. So we use 2, we gain 4, we end up with 2. Okay, so glycolysis happens no matter what, right? So this is the first step to pretty much any biotransformation, if we want to call it that. And so what happens next will shock you. Sorry, a bit of clickbait there. But okay, so now let's talk about the Krebs cycle. So this is also called, as I said, the citric acid cycle, or the TCA. And so this is cellular respiration, right? Then this goes into the electron transport chain after. And so this is all done in the presence of oxygen. Now, fermentation, as I mentioned, is done mostly in the presence of no oxygen, or so in an anaerobic environment, or anoxic. But there are certain situations where we have oxygen present, and there are definitely certain bacteria involved in the brewing process, whether we want them or not, that are aerobic. And so it's good to know about cellular respiration. Amen. Okay. So where were we here? So just for clarification, by the way, redox is basically the key to all biochemical processes. And so it's just important to know what it is. So it's reduction and oxidation, and it's confusing. It's confusing enough that students are taught things like Leo goes grr and oil rig. So respectively, lose electrons, oxidation, gain electrons is reduction. Or oxidation is loss, reduction is gain. You see why it's so confusing, right? So if you gain around the waist, let's say, you don't normally go around saying, well, I used to be a size 38, but I reduced to a size 42. Or, I reduced my grocery bill from 200 a week to 400 a week. So it makes sense from a biochemical standpoint, but it doesn't really make a lot of sense as far as just regular English goes. And so a bit more terminology just to make things a little easier here. So we're going to be talking about NAD plus and NADH. So NAD, nicotiniamide adenine dinucleotide, it's a coenzyme. It's essential to cellular respiration. And then NAD plus is the oxidized version, and NADH is the reduced version. And so acetyl-CoA is another coenzyme. It's acetyl coenzyme A, and this is pretty central to metabolism as well. It kind of serves as a secondary messenger. It's a signaling enzyme. It's just responsible for an absolute ton of different biochemical processes, and it delivers the acetyl groups into the TCA. So it's something also to be aware of here. Okay, let's get rolling. Okay, so the Krebs cycle. Okay, so it's a mitochondrial process. Basically, it uses acetyl-CoA to generate ATP, GTP, reduced coenzyme NADH, and FADH2, and carbon dioxide. And it's an 8-step process. And let's take a look. I just realized that we kind of skipped over one. So FAD is flavin adenine dinucleotide. So FAD is the oxidized form. It's also known as quinone. And then FADH2 is the fully reduced form. And so hydroquinone is also what it's known as. There's another one that exists called semiquinone, but we don't really need to worry about that. So just be aware that it's also a cofactor in redox, and it acts as an electron carrier, sort of the same way as NADH. So just another one for the quiver there. Okay, back to what we were talking about. So step one, so we've got pyruvate, of which we spoke about earlier, and it gets oxidized to acetyl-CoA, and this process generates some NADH and releases a little bit of carbon dioxide. Okay, so starting at the beginning of the cycle, we have this acetyl-CoA, which has 2 carbons, and oxaloacetate, which has 4, and the result of the combination is citrate, which has 6. So then in the second step, this rearranges via isomerization into isocitrate, and then this is oxidized into alpha-ketoglutarate. And that's a 5-carbon molecule, and this releases 1 carbon dioxide and results in NAD plus to being reduced to NADH. Moving on to step 4, it's another oxidative reaction where alpha-ketoglutarate is oxidized to succinyl-CoA, so this is another enzyme, a coenzyme, and this releases carbon dioxide again, and again reduces NAD plus to NADH. And then in step 5, the succinyl-CoA is converted to succinate by moving a phosphate from to ATP or GTP. And then in number 6, the succinate is now oxidized to fumarate. And then in this process, it reduces FAD to FADH2. And then the fumarate is converted to malate via hydration. And finally, the malate is oxidized and regenerates the oxaloacetate, another NADH, and the cycle can begin again. Okay, so it's not very complicated also, but it is. But the Coles notes here are that acetyl-CoA, NAD+, FAD, ADP, or GDP go into the cycle, and then the 2 carbon dioxides, 3 NADH, 1 FAD2, H2, and then 1 ATP are generated and released. And now moving to the electron transport chain, also called oxidative phosphorylation. Essentially, a bunch of molecules pass electrons from NADH and FADH2 down the chain until they reach the oxygen as a final electron acceptor. And so in some cases, as I mentioned, there are oxygen substitutes, but I guess since we broke the seal, we might as well go through this one step by step too. An easy way to kind of think about this is like a fire brigade in the old days before they had, proper equipment. And then you've got, someone filling up a bucket and they pass it to the next person who passes it to the next person, the next person, the next person who throws it on the fire, and then the bucket goes back, blah, blah, blah. So basically the NADH enters the process. NADH dehydrogenase, it's an enzyme. Anything that ends with A's is an enzyme, pulls electrons off, creating NAD+, and releasing energy, allowing protons to be pumped out into the intermembrane. And then the succinate dehydrogenase pulls electrons from FADH2, creating FAD, and this moves these electrons down the chain. And so both of these prior operations move their electrons to coenzyme Q, which carries them to the third molecular complex, cytochrome BC1. And this now pumps protons out again and then moves electrons to the fourth complex, cytochrome C oxidase. This now pumps protons out once again, but half as many. Electrons are then passed to oxygen. This combo with the protons forms water. And then the protons we spoke of were all pumped out into the intermembrane space. This creates what's called an electrochemical gradient. And so this means there's a concentration and a charge difference. And this gradient allows ATP synthase to flow protons back into the matrix and produce ATP. Okay, so I know that none of this helps us get beer, but it's getting closer to the real juice here. So I guess we're going to talk about microorganisms for a second. So bacteria, fungi, and including, of course, yeast, archaea, et cetera, are the microorganisms. And so we can break these, at least for metabolic purposes, or at least for our purposes, into anaerobes and aerobes. Anaerobes don't require oxygen, aerobes do. And within this, there are obligates and facultatives. So an obligate aerobe is dead without oxygen, let's just say. A facultative aerobe finds a way, and this way is fermentation. The cells need energy, as we've established. All cells do. Oxygen isn't there to help in certain situations, or maybe there's really no mechanism with which to use oxygen anyways, if you're an obligate anaerobe. And so the microorganisms break down carbohydrates into byproducts and generate energy. Lactic acid fermentation produces lactic acid, of course. Lactobacillus is sort of the poster child for this. There's a whole bunch of different lactic acid bacterias, LAB for short, but lactobacillus is really the main one we're concerned with most of the time. And that's responsible for sauerkraut, cheese, yogurt, etc. And dill pickles, for example, like fermented pickles, not the ones that are just packed in vinegar. And alcoholic fermentation produces, of course, alcohol, and that's what we're primarily concerned with in this world of beer. We can talk about lactic fermentation down the road too, because it does. Sour beers are a real thing. Okay, so once again, the first step here is glycolysis. So recall this nets us 2 ATP, splits the glucose into 2-pyruvate, and reduces NAD plus to NADH. And this is the same as what enters the Krebs cycle, but it's not going to in this case because there is no Krebs cycle. So the 2-pyruvate via pyruvate decarboxylase are going to turn into 2 units of acetaldehyde and release 2 units of carbon dioxide. And now an enzyme called alcohol dehydrogenase, which is the same enzyme, by the way, that breaks ethanol back into acetaldehyde in our bodies, is going to reduce the acetaldehyde to ethanol using electrons from NADH, oxidizing the NADH back to NAD+, so that the glycolysis can start over again. Lactic fermentation is the same process except with different enzymes, and so in this case, lactate dehydrogenase reduces the pyruvate to lactic acid as opposed to into ethanol, or acetaldehyde and then ethanol. Okay, and so another thing to be aware of is something called the Crabtree effect, which causes yeast to produce ethanol even in the presence of oxygen. And so this happens only in the presence of a glut of glucose. And so fermentation obviously nets fewer ATP molecules per glucose than respiration, but it does allow for rapid cell growth, which is why they seem to favor it. And so this effect was actually first observed in cancer tumors. But the current thinking is that because ethanol is toxic, it's sort of a competition mechanism. And we can get all into this some other time. We'll do a whole thing maybe on Crabtree effect, or we can just talk about it when we talk about yeast. But basically, just know that yeast has the ability, it is facultative, so it does have the ability to produce, to actually go through cellular respiration, but it also has the ability to produce ethanol in the presence of oxygen as long as there's enough glucose lying around. Okay, so that's basically, I think, the overview. We're going to start getting pretty deep into yeast and yeast metabolism, I think. And so, I thought we should go through just the basics of cellular metabolism before we get really deep into the fermentation process. We're going to go into more detail anyways, and I promise not to go through the Krebs cycle again, because I know it's nuts, right? And it's hard even to, for me to talk about it is kind of weird, but it's also even weirder to listen to it. I get that. It's easier to look at a chart. Even those charts don't make a lot of sense half the time. I know the way it's explained in high school biology, nobody understands it, even though it's, like I said, it's not that complicated, but it is like, it's just, I don't know. It's like one of those things. Okay, so I hope that clears up some terminology and some of the processes and makes it a little easier to understand. I don't know if you guys heard that jump there, but I was sitting in a funny way and my oblique cramped up and so I had to pause. Anyways, okay, so where was I here? So I just wanted everyone to understand, what the yeast is basically doing. So the general process of metabolism, as I mentioned. I know I said metabolism about 900 times here, but my side's in a fair amount of pain. I don't know what's going on. Anyways, and various other microorganisms obviously use the same process or processes. And so that's where we're going to leave it for now. So next we're going to go into yeast metabolism, secondary metabolites, and so on and so forth. Until then, stay thirsty, stay curious, and hopefully I make it to the next episode.